So double fertilization. Double fertilization is a characteristic feature of angiosperms. So what happens here in double fertilization? Before you go into double fertilization, you must know about the development of female gametophyte. See, this is what structure of ovule. The ovule has a covering, one or two layered covering called integument. Then the stalk of the ovule is called funicle. Then the integument has a white gap at the top called microphyll. Opposite to the microphyll is the chalasa. Then the integument encloses a mass of homogeneous cells called nucellus. One of the cells in the nucellus functions as megaspore mother cell. So the you would have studied in the development of female gametophyte. Megaspore mother cell. Megaspore mother cell divides by meiosis to form 4 haploid megaspores. In that 4 haploid megaspores, one megaspore becomes functional. The functional megaspore develops into female gametophyte or embryo sac. This is what female gametophyte or embryo sac, this whole structure. Now, how the embryo sac develops in the sense, see, one nucleus in the functional megaspore, it divides into 2. Then they move towards opposite poles. Then again divide further, the nuclei divide further, 4 nuclei. First one, single nucleus, 2 nuclei, move to opposite poles, then divide 4 nuclei. 4 nuclei divide further to form 8 nuclei. Then in the 8 nuclei, 3 nuclei at the microphylla end form egg apparatus. Then 3 nuclei opposite to that form antipodals. 2 nuclei, one from each pole. One from here, one from here. Each pole meet at the center, used to form secondary nucleus. That's what secondary nucleus. So secondary nucleus is a diploid one. Now this is what the development of female gametophyte. That's what embryos are. Double fertilization. Male gametophyte. Pollen grain germinates, and then it produces the time of germination of pollen grain, pollen tube is formed. Then the growth of the pollen tube is controlled by tube nucleus. Then each pollen grain produces 2 male gametes. In the 2 male gametes, one male gamete fertilizes the egg, resulting in the formation of diploid zygote, haploid plus haploid, resulting in the formation of diploid zygote. Zygote develops into embryo. Another male gamete fertilizes the secondary nucleus to form primary endosperm nucleus. The primary endosperm nucleus develops into endosperm, which is a triploid one. Why triploid? Secondary nucleus is formed by fusion of 2 polar nuclei, diploid one. This one haploid, 2n plus n, 3n. It is called triple fusion. Why it is called triple fusion? In the sense, see, 3 nuclei fuse. Which are the 3 nuclei? 2 polar nuclei, one from here, one from here. 2 polar nuclei. Then the male gamete, 1 nuclei. The 3 nuclei fuse. That's why it is called triple fusion. Double fertilization and triple fusion. Now, after this, you will have to look into the post-fertilization changes. What are the changes taking place after fertilization? See, after fertilization, you know very well about calyx, corolla, then stamens, style. So these structures wither and fall off. Now about the ovary, you know very well about the structure of gynoecium. Ovary, style, stigma. Ovary develops into fruit. In that ovary wall becomes pericarp, ovules become seeds. Then microphyll becomes microphyll of the seed. Microphyll of the ovule becomes microphyll of the seed. Opening of the seed. Then integument becomes seed coat. So what is the seed coat? The outer integument is called, develops into outer seed coat, namely testa, hard layer. The inner integument develops into tegmen, thin layer called tegmen. Now nucellus, sometimes nucellus develops into perisperm. Then next one about zygote. Zygote develops into embryo. The secondary nucleus develops into primary endosperm nucleus, which gives rise to endosperm. What happens to antipodals? Antipodal synergies degenerate, disappear. They degenerate or disappear. That's what you are going to see in the post-fertilization changes. Next one. Post-fertilization changes. Pause before fertilization, after fertilization. Actually, the transformation after fertilization. Sepals, petals, stamens and stigma wither and fall off. Ovary becomes fruit. The ovary wall becomes pericarp. Ovules become seeds. Ovule become seeds. Then egg develops into zygote. Egg is the haploid one. Zygote is a diploid one. Zygote develops into embryo. Then funicle, stalk of the seed. Microphyll becomes microphyll of the seed. Nucellus becomes perisperm. Outer integument develops into testa. Inner integument becomes tegmen. Then synergies and antipodals degenerate. Secondary nucleus develops into endosperm. So, secondary nucleus becomes primary endosperm nucleus, which gives rise to endosperm. So, these are the post-fertilization changes. Sepals, petals, stamens and stigma wither and fall off. Then ovary becomes a fruit, ovules become seeds, egg develops into zygote, funicle becomes stalk of the seed. Then microphyll of the seed, nucellus becomes perisperm, outer integument testa, inner integument tegmen. Synergies and antipodals degenerate, secondary nucleus gives rise to endosperm. So now you must know about the endosperm. What is endosperm? When it develops endosperm? So the endosperm is a nutritive tissue. Endosperm is a nutritive tissue which supplies nutrition for the developing embryo. For example, you know very well about the coconut. See, in the coconut, hard layer, actually the fruit is a drupe, the hard endocarp. So that you studied last year. Now when you just look into that, here you get white structure. So that white one is the endosperm. So what kind of endosperm? This endosperm, cellular endosperm. Then of course you get liquid. Liquid nana இளநீன்னு சொல்லலாம் அல்லது தேங்காய் தண்ணின்னு சொல்லுவோம் இல்லையா உள்ள இருக்கிற தண்ணி அதுதான் அது வந்து it is also endosperm, but what kind of endosperm? It's a liquid endosperm. So this is what you can just see in the coconuts. Where you come across this. Now endosperm. See, endosperm, how it develops endosperm? Secondary nucleus. In the ovule, the structure of the ovule, inlet, integument. Here you get egg apparatus, secondary nucleus, antipodals. So male gamete fertilizes the secondary nucleus to form primary endosperm nucleus. The endosperm develops into the primary endosperm nucleus. This one secondary nucleus. Secondary nucleus, then male gamete, another male gamete, second male gamete, fertilizes the second nucleus to form primary endosperm nucleus. So the primary endosperm nucleus gives rise to endosperm. Normally endosperm is a triploid one. Why triploid? Secondary nucleus is diploid one. Male gamete haploid one, haploid plus diploid, you get triploid one. So endosperm, primary endosperm nucleus divides to form an endosperm. The endosperm develops, when endosperm develops before the development of zygote. Development of zygote in the sense, zygote develops into embryo, before the development of embryo. Actually, before the division of zygote in the sense, before the development of embryo. Endosperm develops before the development of embryo. before the division of zygote. So that's what division of zygote. Zygote divides into embryo. This zygote divides to form embryo. So even before that, endosperm develops. Why endosperm develops? It supplies nourishment for the developing embryo. Endosperm supplies nourishment for the developing embryo. So that you can see in the coconut again. So in the coconut, white purple சொல்லுவாங்க. அது வளர வளர என்ன ஆகும் அப்படின்னா அந்த எண்டோஸ்பெர்ம் எல்லாத்தையும் சாப்பிடும். அதுக்கப்புறம் பார்த்தீங்கன்னா உள்ள ஒண்ணுமே இருக்கு. பிளான்ட் வந்துடும். அந்த எண்டோஸ்பெர்ம் சப்ளை பண்ணி தான் இது பண்ணுவாங்க. பிளான்ட் டெவலப். So the embryo grows only by the usage of endosperm. Endosperm supplies nourishment for the developing embryo. That's why endosperm supplies nourishment for the developing embryo. So that you will study in the functions of embryo. Now, types of embryo. Embryo is of types of endosperm. Endosperm is of 3 different types. Based on the development, endosperm is of 3 different types. Cellular endosperm, nuclear endosperm, and then helobial endosperm. Nuclear endosperm, cellular endosperm, and helobial endosperm. So, first one about nuclear endosperm. So, you just see that here, this is one endosperm nucleus. Single nucleus. The single nucleus divides into many. Endosperm nucleus is a triploid one. So divides into many to form many nuclei. So all the nuclei are triploid one. So this kind of endosperm is called nuclear endosperm. Division of nucleus is not followed by wall formation. That's a free nuclear division. So single nucleus. What kind of nucleus? Primary endosperm nucleus. The primary endosperm nucleus divides continuously without wall formation, resulting in the formation of what kind of endosperm? Nuclear endosperm. Example: coccinea, capsula, and arachis. Arachis groundnut. Primary endosperm nucleus undergoes several mitotic division. Primary endosperm nucleus undergoes several mitotic divisions to develop free nuclei. That's what single nucleus divides into many by mitosis to form what? Nuclear endosperm. Example: coccinea, capsula, arachis, hypogaea. Then cellular endosperm. See, in the cellular endosperm, what happens here? Single primary endosperm, and then it divides into two. It divides into two into many cells. So only cells are formed. 2 into many cells. So in the set again 2 into 4 cells. 4 cells like that divides into many. So this kind of the nuclear division is followed by wall formation. Many cells are formed. That kind of endosperm is called cellular endosperm. Example: Doxa, Helianthus, Scoparia. So Helianthus, sunflower. Helianthus is the sunflower. Helobial endosperm. So quite different one. Helobial endosperm. In helobial endosperm, the primary endosperm nucleus goes to primary endosperm nucleus moves towards the lower end and it divides into two from larger portion on the upper side. So divides into 2 nuclei followed by wall formation. Divides into 2 nuclei. Single nucleus moves downwards and divides into 2, and then it is followed by wall formation. So in that wall, it results in the formation of large cell at the microphylar end and small cell at the chala cell end. Microphylar end, chala cell end. Now this the largest this cell, the microphylar chamber divides the nucleus cell. The nucleus divides into many to form many nuclei. In the lower side, it may undergo division or it may not undergo division. This kind of endosperm is called helobial endosperm. Example: Hydrilla, Vallisneria. These are aquatic plants. Okay, 3 types of endosperm. First one about nuclear endosperm. Single primary endosperm nucleus divides into many. The nuclear division is not followed by wall formation. What kind of divisions? Repeatedly mitotic divisions. It is not followed by wall formation. That kind of endosperm, nuclear endosperm. Okay, example: coccinea, capsula, arachis. Then next one, cellular endosperm. primary endosperm, primary endosperm nucleus, only one cell. It divides into two. It moves towards lower end, divides into two, and then followed by wall formation. So larger portion at the microfilar end, smaller cell at the chala cell end. The larger portion, microfilar region, the nucleus divides into many to form many free nuclei. Then the lower end, it may undergo division or may not divide. It may undergo cellular division or may not divide. This kind of endosperm is called kilobial endosperm. Example, Hydrilla vallis media. So these are the three types of endosperm. The next one about endosperm. The endosperm may be ruminate. Ruminate in the sense the endosperm forms in the form of layers. Endosperm is formed in the form of layers called ruminate endosperm. So example, betel nut, myristica. Betel nut and myristica. What it is myristica? Betel nut, Areca catechu, myristica fragrance, myristica, ஜாதிக்காய்னு சொல்றோம் இல்லையா, அந்த ஜாதிக்காய் மேலே வந்து இந்த மாதிரி ஒரு லேயர் இருக்கும், அது மேலே இப்படி படிஞ்சு இப்படி இருக்கும், அதுதான் பத்திரின்னு சொல்லுவாங்க. அதுதான் அது வந்து myristica fragrance. So that is what ruminate endosperm. Here இது உள்ள பார்த்தீங்கன்னா the seed myristica, ஜாதிக்காய் உள்ள பார்த்தீங்கன்னா very லேயராக இருக்கும். So that's what called ruminate endosperm. Now the seeds with endosperm are called endospermous seeds or albuminous seeds. Endospermous seeds or albuminous seeds. Example for that, castor, paddy, coconut, endospermous seeds. And then seeds without endosperm are called exalbuminous seeds or non-endospermous seeds. Seeds without endosperm are called non-endospermous seeds or exalbuminous seeds. Example, beans, peas and groundnuts. So there are one more question. The next one, functions of endosperm. Endosperm, it gives nutrients to the developing embryo. That's why it supplies nourishment to the developing embryo. Nutritive tissue for the developing embryo. It regulates mode of development of embryo. Then zygote divides only after the development of endosperm. So that's what endosperm develops even before the division of the zygotes. That's what given here. Okay. Then next about development of embryo. So in the development of embryo, only few things given to you, not given in a detailed account of that one. Zygote is a diploid cell. Zygote divides transversely. Zygote divides transversely into lower cell and terminal cell. This one terminal cell, this one is lower cell. So and then next one, the terminal cell divides. The terminal, and then the 2-cell embryo divides into 4-cell embryo, then 8-cell embryo. Then it gives rise to embryo. So in that embryo, which all the structures you get in the sense, in the embryo you get plumule, radicle, then suspensor, plumule, radicle, suspensor, cotyledon. So this is what about the development of embryo, but not given in a detailed account of that one. Zygote is a diploid cell. So the zygote divides by mitosis into 2 cells. Actually, which is side in the sense, terminal cell towards microfilar end, and then lower cell towards the chala cell end. And then now the upper cell divides into 2, and then lower cell becomes suspensor cell. Another divisions to give suspensor cell. Well, you would have studied about the suspensor, role of suspensor in gymnosperms. Suspensor pushes the embryo deep into the female gametophyte. Then after that, what happens here? The 4-cell embryo, then global embryo. It gives rise to mature embryo. In the mature embryo, which are the structures you get? Cotyledons, plumule, hypocotyl, epicotyl, radicle, root cap, suspensor. Role of suspensor: suspensor pushes the embryo deep into the female gametophyte. Suspensor pushes the embryo deep into the nutritive tissue. Suspensor pushes the embryo. That's all. Suspensor pushes the embryo. Whereas in gymnosperm, it pushes the embryo deep into the female gametophyte. Okay. Zygote is a diploid cell. How zygote is formed? Zygote is formed by the fusion of male gamete with the egg. Then egg actually male gamete fertilizes the egg, resulting in the formation of diploid zygote. So during the development of embryo, what happens? Zygote divides into two. Upper cell called terminal cell, lower cell called basal cell. And then the upper cell divides repeatedly to form embryo. What kind of embryo? Heart-shaped embryo. First we have given globular embryo and then heart-shaped embryo. Then in that embryo, the embryo consists of plumule, radicle, cotyledon. Plumule, radicle and cotyledon. So that further things you will see in the next class.